next speaker is Dr. William Decker, also from the Medical Center in Houston. Dr. Decker, his educational background is bachelor's in biochemistry from U of H, University of Houston. He has a bachelor's of science from Tufts as well. He received his PhD from Baylor College of Medicine, also in Houston, Texas. He has a doctorate in molecular and human genetics from that institution. He is at MD Anderson Hospital and various research laboratories, and is working on regulatory pathways and so forth that relate to stem cell therapies. He's thoughtfully on progress in clinical applications. Dr. Decker.
Okay. I'd like to thank the organizers for letting me come here today and talk to you. Originally, I was told this is going to be a CME talk. I don't have any data to show you. However, I am going to try to present my opinion of the cell therapies that are in the laboratory today or are in early stages of clinical development today that you will be seeing hit the clinic in the next decade and that you will want to use yourselves in your practices. And this is my disclosure. Pay attention, it's meaningful. My principal interest is in the immunotherapy of cancer. I spent the last decade, and will probably spend the next decade, unlocking secrets of immunotherapy so that we can address cancer in a meaningful fashion. The history of immunotherapy for cancer is very old. It goes back over 100 years to the work William B. Coley did in the 1890s. Between William B. Coley's work and the modern era, it was thought that cancer could not be appropriately addressed by the immune system. And a lot of basic work had to be done over the intervening 60, 70, 80 years before people realized indeed that the immune system was very important in the treatment of cancer. And this work took a long time and was the culmination of results from many different investigators. Initially, it had to be discovered that tumors indeed were immunogenic, meaning that they could be recognized by the immune system as foreign. Scientists and researchers also had to learn exactly how tumors talk to the immune system, so that the immune system could indeed figure out that there were parts of cancer that might be seen as foreign. More work in the 1970s by Ralph Steinman led to the discovery that dendritic cells are the principal antigen-presenting cells of the human body and are responsible for directing the immune system to attack cancer when it does so. And since that time, there's been a tremendous amount of work done on dendritic cells, 30, 40 years of work, in order for us to arrive at the point where we are today with a bunch of cancer vaccines that don't work. However, they don't work for different reasons than they didn't work 30 or 40 years ago. So there are several cell-based immunotherapies that I'm going to talk to you about today. One I've just spoken to you about is dendritic cell vaccine therapy. The other two are adoptive therapies using the immune cells that actually go after, recognize, and kill cancers. Those two types of cells are T cells and NK cells. So what exactly is a dendritic cell? The dendritic cell is the principal cell of the mammalian immune system that is responsible for directing the activities of the immune system. It tells all of the other cells what they should recognize, what they shouldn't recognize, and what they should do. Your bodies are full of dendritic cells, and they're constantly sampling all of the different proteins and molecules and antigens that are constantly passing through you. And by and large, they do nothing, if told not to do anything. When a dendritic cell senses danger, it rapidly samples all of the antigens, proteins, and molecules in the area where it is, and then it migrates to the nearest lymph node where it will direct the immune system to attack. Typically attack those cells that are expressing the antigens, molecules, or other proteins that it has sampled when it sensed the danger signal. So dendritic cell immunotherapy which has been going on now for 15 years and will continue to go on, consists of the loading of your own dendritic cells with what we call cancer antigens. What's a cancer antigen? A cancer antigen is a protein, essentially, that can be used to differentiate a cancer cell from a normal cell. And all cancer cells have them, and they tend to be patient-specific. Once dendritic cells have been loaded with these cancer-specific antigens, we treat the dendritic cell with a variety of different chemical or biologic stimuli to cause what we call a Th1 response. A Th1 response simply is an immune response that is cell-mediated. We take these reconditioned dendritic cells and we put them back into the patient and let them talk to the patient's immune system and attack the cancer. Many people in the room may be aware of Provenge. That's a recent cancer vaccine which is based upon this principle, the introduction of a specific cancer antigen to the patient's own dendritic cells, and then the reintroduction of those dendritic cells back into the patient. This was recently approved for use in stage 4 asymptomatic patients with carcinoma of the prostate. And it imparts, on average, an extra 4 months of life. However, that doesn't tell the whole story. And the fact that it was approved by the FDA is highly significant, not just for the company that makes it, but also for the field in general. I'm going to switch gears now and talk a little bit about the immune cells that actually do all the work. First are the T cells. These are the actual cells in the body that are responsible for discerning self from non-self, foreign from sane. And they recognize and eliminate cells that are displaying foreign antigens on their surface. And as I told you previously, their function is orchestrated and controlled by the dendritic cells and other immune molecules. So in adoptive T cell therapies, which is something that we're very interested in at the MD Anderson Cancer Center, T cells are trained outside the body to recognize a certain antigen or a certain suite of antigens that might be specific to a particular cancer. Then grow up these T cells, we multiply them by thousands and millions of folds outside the body in a tissue culture flask, and then we reinfuse them into the patient. There are also some adoptive therapies whereby these T cells are actually genetically manipulated so as to recognize foreign antigens of interest, and again, reintroduced into the patient. There are a wide variety of these trials going on right now, and some have shown some reasonable promise. So this is another therapy you can expect to see more and more of in the coming decade. I'm going to talk to you about a second type of immune effector cell called a natural killer cell. A natural killer cell is a little different from a T cell. T cells recognize cells in the body that are displaying foreign antigens. One thing that cancers are very good at is downregulating or eliminating the machinery that displays foreign antigens. So that's where natural killer cells come in. They're constantly scanning the cells inside the body, looking for cancer cells or other cells that are downregulating the machinery that allows T cells to interact with them. And as it turns out, there are a wide variety of cancers that do this. So we can take natural killer cells, grow them up in very large numbers, and infuse them back into the patient to go after many different kinds of cancers, specifically hematopoietic cancers like leukemia or lymphoma or myeloma, and those are the kinds of active projects we have ongoing right now. So to put some technical terms to what I just said, natural killer cells eliminate cells inside the body that exhibit low levels or absent levels of MHC class 1. MHC class 1 is the molecule of the cell responsible for presenting antigens to the immune system. NK cells are activated and suppressed by a very complicated mechanism of activation and inhibitory signals. The most important one here is the killer cell immunoglobulin-like receptor, which is also called a KIR. when we give NK cells to a patient, you'll hear the term KIR mismatching. And that simply means the use of third-party NK cells that have a different system of KIRs than the patient, so that there's absolutely no chance that the patient's own cells can inhibit these NK cells. They will go in and they will do their job on cancers that are downregulating MHC class 1, and there's no opportunity for them to be inhibited. Which again is what I just explained. So those are the three types of cell therapies that we're using in cancer and that will likely make important impacts in the clinic in the coming years. Also, in my capacity as a consultant, get to do a lot of work with stem cells, or at least a lot of theoretical work. And the MD Anderson Cancer Center itself is also starting a new stem cell center in which we're going to be looking into the uses of many different kinds of stem cells. So I thought I'd talk about stem cell therapies as well, because those are also going to be making a big impact in the clinic in the next 10 years. In the field of regenerative medicine, as just about everybody's talked about so far, we use stem cells, which again can include a wide variety of different cell types, to regenerate damaged, destroyed, or missing tissues. Stem cells can include what some have talked about today, autologous, which means from the same being, adult stem cells, i.e. cells that come directly from the patient, or pluripotent stem cells, which can come from a variety of sources. Pluripotent simply means a stem cell that has the ability to regenerate every cell type and tissue of the human body. And as you're going to see, not all of these cells need necessarily come from an embryo. So again, we've talked a lot about the applications of adult stem cells today. I'll just add a little bit more. At the MD Anderson Cancer Center, we use hematopoietic stem cells for transplant, for bone marrow transplant. When somebody has leukemia and has reached an advanced stage and is dying, we can often give a stem cell transplant to save that person's life. Hematopoietic stem cells are a good example of adult stem cells. They can only make tissues of the blood. Mesenchymal stem cells have been talked about a little bit too. We use those for several applications as well. We can use mesenchymal stem cells as a feeder cell layer to expand hematopoietic stem cells, so as to allow more rapid engraftment when patients receive a stem cell transplant. Mesenchymal stem cells can also be used to reconstitute bone, to repair large bone defects that some patients might experience. They can be used for ex vivo tissue regeneration, and they can also be used for immunoprotective therapies. There are a wide variety of uses for this cell type. In practical terms, people have used adult stem cells, specifically muscle stem cells called myoblasts, to regenerate skeletal or cardiac muscle. And investigators have also used corneal stem cells to regenerate damaged corneas. And there are, of course, a variety of other examples, all of which you've already heard about today.